Hello dear students. The topic of today's discussion is palynology in relation to taxonomy. So what do you mean by palynology? Palynology is a science which deals with the study of pollen grains and spores. Pollen means it is the male gamete of a plant. Pollen grains are the male gametes. They have tough sporopollen in the outer coat, so they are well preserved even in fossils. The term palynology was coined by two people, Hyde and Williams, in the year 1944. So palynology literally means the study of dust. It is derived from Greek words. Palynine means to scatter. These are a few pictures to show you the exact position of the pollen grains. You can see a flower of hibiscus and the stamens from which the pollen grains, the spinous pollen grains of hibiscus. The top right is that of a stamen, filament and anther. It shows filament and anther. And the next diagram is that of a transverse section of anther, tiers of anther, showing pollen grains and line of dehiscence. Then the down left picture is that of the tiers of anther through a microscope. You can see 4 pollen lobes, and inside them are the pollen grains, pollen sacs and pollen grains. Then the left bottom picture is that of a single pollen grain. It shows 2 walls, a continuous intine and a discontinuous exine. Exine has certain thin areas which are pits or germ pores through which the intine comes out in the form of a tube called pollen tube, and inside is the 2 nuclei, a generative nucleus which later divides to form 2 male gametes, and then the pollen tube nucleus or the vegetative nucleus. There is a very thick exine which has a outer sculptured wall. Now the pollen characters of taxonomic interest. The following characters must be studied in order to know the taxonomy of certain plants. The size of the pollen, the polarity of the pollen, which is the distal pole and which is the proximal pole, the shape of the pollen, what is the shape of the pollen, the symmetry of the pollen, how is it symmetrical, the number and type and arrangement of apertures of the sporoderm stratification and patterns of ornamentations are studied, and the physicochemical properties of the pollen walls. All these external features of pollen grains are studied, and all of these form important for plant classification. Now first of all, coming to the size. To discuss about the size of the pollen grains vary from less than 10 microns to more than 200 microns. Less than 10 microns is very less, 10 to 25 microns is small pollen grains, 26 to 50 microns is median, 51 to 100 is large, 101 to 200 is very large, and more than 200 is gigantic pollen grains. Coming to the polarity of pollen grains. When pollen grains are in tetrads, the portion which comes in the center is called the proximal pole or near the point of attachment. The side facing the point of attachment of 4 pollen grains is called as the proximal pole, and the position opposite to the proximal is called as the distal pole or the pole which is in distance or away from the point of attachment of the 4 pollen grains. The line joining the 2 poles is called as the polar axis, and the line at right angles to the polar axis is equator line or equatorial axis. In this figure we can see the tetrahedral condition of the pollen grain. when they get separated from tetrad, we cannot distinguish the proximal pole and the distal poles. And isopolar condition is, so such indistinguishable condition is called as apolar spore. Cryptopolar spores are those which require closer examination. The polar spores are conspicuously distinct. There are two types, isopolar and heteropolar. Isopolar ones are the ones in which the two poles are exactly similar. Heteropolar pollen grains are those in which the two poles differ. There is an aperture in the distal pole but no aperture in the proximal pole. Subisopolar pollen grains, the distal and proximal poles, there is very little difference in the distal and proximal poles. Cryptopolar pollen grains, close observation or examination is needed, and the distal pole and proximal poles are dissimilar. There is difference in sculpturing and in the polar and the distal poles. Coming to the shapes of pollen grains, many shapes are seen in pollen grains. Radiosymmetric, the radially symmetrical ones are called radiosymmetric, and they can be ellipsoidal, circular, disc-like, spherical, etc. Paroblate, when the polar and equatorial axis is less than 4/8. Oblate, when the polar and equatorial axis is 5/7. Subspheroidal, when pollen grain is sphere-like but pressed on one side. Perprolate, the polar axis by equatorial axis is more than 8/4. Here we can see the pollen shape classes with their ratios. The PA divided by ED into 100. This is the formula with which we calculate the pollen shape classes. When we calculate in this formula, we get these following shapes: paroblate, oblate, suboblate, oblate, spheroidal, spheroidal, prolate, spheroidal, subprolate, prolate, perprolate. So these are different shapes of pollen grains on the basis of polar and equatorial axis. Polar axis upon equatorial diameter into 100 gives all these above shapes. Then coming to the symmetry of the pollen grain. Asymmetric pollen grains are those which can't be divided into two equal halves in any plane, and these are rare. Symmetric pollen grains can be divided into two equal halves through one or more planes. Symmetric pollen grains are again of two types: radiosymmetric and bilaterally symmetric. Radiosymmetric are those in which two equal halves can be done through more than two planes. Bilaterally symmetric ones are those in which two equal halves can be got from one or at the most two planes. So these are a few diagrams, pictures of the pollen grains. Next, coming to the apertures. Apertures are those areas in which there is no exine deposition. The intine inner pollen wall is continuous. Above the intine there is exine wall material deposited. Here and there exine is thin at one or more places, and such places are called as germ pores or apertures. The pollen with apertures are called as aperturate pollen, and pollen with no apertures are called as non-aperturate. Exine is uniformly thick, leaving no gaps or thin areas. Aperturate pollens differ in position, shape, size, and number of apertures. Position of apertures, shape of apertures, size of apertures, and number of apertures. First of all, we will discuss about the position of apertures. Apertures can be polar, global or equatorial. Polar can also be monopolar or bipolar. Polar means the apertures are at the poles, and they can be either monopolar when there is only one aperture at one pole, which may be either proximal or distal. Bipolar when there are two apertures on both the poles. Global pollen grains are those in which there are apertures on all the walls of the pollen grain. Equatorial apertures are apertures on the equatorial region of the pollen grain. Coming to the shape of the aperture. Shape can be round or elliptical depending upon their position as given below. Laesura, at the proximal pole, a single narrow elliptical aperture. Sulcus or furrow, single narrowly elliptical aperture at distal pole. Colpus, plural colpi, one or more narrowly elliptical apertures in the equatorial plane. Colpate pollen grain. So, lacerate pollen grain, sulcate pollen grain and colpate pollen grain. Lacerate pollen grain is when there is a single narrow elliptical aperture at the proximal pole. Sulcate pollen grain is one in which there is a single narrowly elliptical aperture at the distal pole. Colpate pollen grains are those in which there are one or more narrowly elliptical apertures in the equatorial plane. So these are the pictures. The pollen wall or exine is divided into sexine and nexine. The sexine, in the sexine if there is an aperture, it is ectoaperture. In the nexine if there is an aperture, it is endoaperturate. Aperture can be circular in the equatorial view, or aperture if it is elliptical, it is colpus. The deep diagram in the equatorial view. Colpus in equator, the third diagram, is called as sulcus. Narrow elliptical aperture at the distal pore is called as sulcus. So this is the polar view which we are seeing in the E diagram. Distal pole, there is a narrow elliptical aperture. So in the polar view, at the proximal pole if there is an aperture, it is an ulcerate pollen, ulcus. That aperture is called ulcus and the pollen is ulcerate pollen. Then you can see monolete spore. In the equatorial view, there is one fissure. You can see one fissure. Now this is the NPC system of classification based on Gunnar Ertmann, who proposed this system in 1969. This is based on the apertures. This single diagram describes the NPC system of classification. So the NPC is number, position and character of apertures. The classification of pollen based on number, position and character of apertures. Based on the number, there are 9 groups of pollen grains. Based on the position, there are 7 groups of pollen grains. And based on the character of the aperture, there are 7 groups of pollen grains. Based on the number of the apertures, 9 groups. Position of apertures, 7 groups. Character of apertures, 7 groups. Now first of all, coming to the number of the apertures. Classification of pollen grains based on the number of apertures. First one is atrium. A is no, means there are no apertures. A pollen grain without any aperture is called atrium. Excuse me. Then a monotreme. Monotreme is N1. It has got only one aperture. Then ditreme, 2 apertures. Tritreme, 3 apertures. Forget about the position and character of apertures. This is solely, here we will concentrate on the number of apertures. N4, tetratreme, pentatreme, N5, hexatreme, N6, polytreme, N7. If it is 7 or more, it is polytreme. Anemotreme, N8. Irrespective of the number, there are many irregularly arranged spiral apertures. In the form of a spiral manner, apertures are arranged. Many number of apertures are arranged in the form of a spiral manner. Then coming to the position. Based on the position, there are 7 groups, which is P0. There is no position for the no apertures. Catatreme, P1, aperture on the proximal face. Position of the aperture is proximal. On the proximal face, if there is an aperture, then it is catatreme. Anatreme, P3. In the distal pole, if there is an aperture, position of the aperture is distal, then it is anatreme or P3. Anacatatreme, P2. There are 2 apertures both on the proximal and distal face. Then it is called anacatatreme or P2. Zonotreme, P4. P4 is equatorial region. If there are apertures, then it is zonotreme and it is designated as P4. Dizonotreme, 2 rows of equatorial apertures. Apertures on the equatorial region in 2 rows. Dizonotreme, it is P5. Pantotreme, globally all over the pollen surface, if there are apertures spread over uniformly, then it is P6 or pantotreme. This classification is solely based on the position of the apertures. Then coming to the character of apertures. Based on the character of apertures, there are 7 groups. It is designated as C. C0, aperture is unknown, there are no apertures. C1, thin area or leptoma, only one thin area. A thin area is the character. One thin area, C1, which is leptoma. It can be monoleft or cataleft or analeft. Monoleft is single thin area, cataleft and analeft in the proximal and distal portions. Then C2, it is called as trichotomonocolpate. 3 slits, very thin, like colpus. C3 is colpate, a single furrow in the center, and C4 is porate, a pore or an aperture in the center. C5 is colpa and pore both present as an aperture. If colpa and pore both are present as an aperture, then it is called as C5 or colporate. Pororate, pore and aura both are present. C6, it is denoted as C6 and called as pororate. Pore and aura both are present. Here we can see monolete and trilete types. Monolete is one fissure. One fissure, if there is one fissure, then it is monolete. If there are 3 fissures, then it is trilete. If there are several fissures, then it is polyplicate. Several fissures, polyplicate. If there are air sacs, vesiculate. Sacs are present, saccate. If there are no apertures, inaperturate. If there is one colpus, monocolpate. If there are 2 colpi, dicolpate. 3 colpi, tricolpate. Full equatorial region has colpus, zonocolpate. Colpe all over the surface. If there is uniform distribution, then pantocolpate. If there is 1 pore, monoporate. 2 pores, diporate. 3 pores, triporate. Equator pores, zonoporate. Uniformly all over, pantoporate. If there are window-like spaces on the pollen where tectum is absent, it is called as fenestrate. Fenestrate. Colpus and pore both are present at 3 places, tricolporate. Tricolporate, both colpus and pore present at 3 places. Zonocolporate, zonal or equator, both colpus and pores are present. Both colpus and pore in the zonal or equatorial region, it is called as zonocolporate. Syncolpate, two or more than two colpi and their ends are joined. Sometimes they are in a spiral form. Then they are called as syncolpate. And you can also see dyads and tetrads. In the right side figure, we can see the dyad, tetrads and polyads. Microsporogenesis yields 4 microspores which mature into 4 pollen grains. In majority of angiosperms, the pollen grains separate prior to release. Such pollen grains are known as monads. In many pollen grains, the pollen grains do not separate and form tetrads. There are 5 different types of tetrads which are identified in angiosperms. Tetrahedral tetrad in ericales, linear tetrad in Typha genus, rhomboidal tetrad, 2 separated by close contact of the other 2. Rhomboidal tetrad. And then we can see the polyads in Calendra of Mimosoideae, 8 to 10 pollen grains in a group. And then in Calotropis and Pollinia, Calotropis and orchids, we can see Pollinia. That is, a pollen grain of a single theca is all fused together to form a cluster. It is called as Pollinia. Next is sporoderm. The spore wall of microspores is in many layers and it is called sporoderm. So, spore coats are collectively known as sporoderm. Sporoderm has 2 layers, sclerine and intine. Sclerine is further differentiated into perine, which is the outermost layer, and then there is exine. Exine is again divided into sexine and nexine. Sexine is differentiated into ectosexine and endosexine. Nexine is differentiated into ectonexine and endonexine. Endonexine, ectonexine and perine form the sculptine. Here we can see the sexine pattern. There are 2 types of exines based on the sexine pattern. They are pilate exine and tectate exine. So the sexine consists of stalked bodies with swollen tips. Each is known as pilum. The stalk and head of pilum are known as caputa and baculum. Stalk is called as baculum, head is called as caputa. The caputa gives rise to ectosexine and the bacula to endosexine. The thin membrane covering the caputa is called as tegillum, Tegillum. In tectate exine, tectum is roof-like, covered with no gaps. In nexine, there are 2 layers, N1 and N2, and innermost layer of spore coat is intine. This is pollen sculpturing. Based on the sculpturing of the exine, these many types of pollen grains can be differentiated. Baculate, clavate, spinulose, foveolate, reticulate, fossulate, verrucate, gemmate, ciliate, striate, areolate, rugulate, perforate, etc. Then the LO analysis. Pollen grain is round and not flat. LO analysis is the shades of microscopical patterns and their variations at successive adjustments of microscope, or it is also defined as the study of pollen grains in transmitted light of the different island patterns produced by solid and non-solid details in successive sporoderm layers is known as LO analysis. Both sporoderm pattern and LO analysis give plant relations, similarity and dissimilarity, and they help us to know the correct systematic position and even up to the species level. Now coming to the evolutionary sequences of angiosperm pollen. The fossil spores are found in peat and sediments. So they are evident since the Precambrian times, that's hundreds of millions of years ago. Brenner in 1996 gave a model of evolutionary sequence of angiospermic pollen. The earliest pollen are small, circular, tectate, columellate without apertures. Gradually with evolution, there was thickening of intine and thick endexine, and sulcus started developing. Then monosulcate pollen grains were seen. After that, tricolpate ones came into existence. Lastly, monocolpate and multiporate pollen grains evolved. Now these are some examples of palynological studies. The primitive dicots and monocots had monocolpate pollen grains. Anemophilous pollen grains are small, round, smooth, thin-walled, dry, with shallow furrows. Example, Populus, Poaceae, Cyperaceae, Betulaceae, etc. All the wind-pollinated ones. The insect or bird-pollinated pollen grains have large sculptured adhesive coating of wax or oily substance. Magnolidae has binucleate pollen. Caryophyllaceae has trinucleate pollen. Ericaceae have tetrads. Asclepiadaceae have pollinia. Quercus has got very small. The pollen wall is scabrate. Linaceae and Plumbaginaceae have similar pollen, which are zonotreme, tricolpate, or pantotreme. Nepenthaceae and Droseraceae have spinuliferous pollen tetrads, except in Drosophilum. Polygalaceae and Ephedraceae have similar pollen grains. In Phytolaccaceae, Phytolacca has 3 zonocolpate and Rivina is pantocolpate. There are 7 genera of Polygonaceae which differ in pollen morphology. All the 7 genera of Polygonaceae differ in their pollen morphology. Then in Salicaceae, there are long, narrow, 3-furrowed pollen grains in Salix and inaperturate spherical pollen grains in Populus. In the different species of anemone obtusifolia has 3 zonocolpate, anemone alchemellifolia has pantocolpate, anemone fulgen has spiroperturate pollen. Podophyllum has united pollen, and so it is separated from Berberidaceae. In Malvaceae and Asteraceae, the pollen has got spinous exine. In Plumbaginaceae, exine is verrucate. In Poaceae, exine is smooth and sulcate. On the basis of palynology, many families have been given their exact position. Fumariaceae has been separated from Papaveraceae. In Nelumbonaceae, Nelumbo has been separated from Nymphaeaceae. In Malvaceae, there is spinous exine, and in Bombycaceae, there is reticulate exine. So, on this basis, they are separated. In Magnoliaceae of dicots, we can see monocolpate pollen grains, and in Ranunculaceae, tricolpate pollen grains are seen, and so they are separated as 2 distinct phylogenetic stalks. Monocots are considered close to Magnolian stock because of its monocolpate condition. Magnolian dicots are considered primitive to Rennelian dicots. Kuprianova in 1948 suggested that most monocots evolved from Ericaceae or Liliaceae. Thus, palynology Thus, to conclude, palynology in relation to taxonomy is the study of pollen grains. Fossil spores are found in peat and other sediments, in lignite, coal and shales. Application of such findings proved invaluable for subsequent studies of ancient climate, particularly the glacial and interglacial stages. Palynology is also invaluable to evolutionary and taxonomic research and can help to delineate phylogenetic relationships between fossilized and extant plants. Pollen grains morphology also plays an important role in taxonomic classification. Thus, we know how the study of pollen grains is helpful in taxonomy. Thank you.